Hi dear students. So in this session, we are going to discuss about mold and yeast forms. So what are fungi? Fungi are eukaryotic microorganisms and it consists of two major groups. The multicellular filamentous fungi termed molds and the unicellular fungi termed yeast. The study of fungi is termed as mycology and the study of fungal diseases is termed as mycosis. All molds are fungi, but all fungi are not molds. Yeast are fungi, but they are unicellular and produce no aerial mycelium. Molds are filamentous fungi that produce aerial mycelium. The kingdom of the fungi can be divided broadly into three groups, that is yeast, molds and the macroscopic fungi. The macroscopic fungi includes mushrooms, bracket fungi, stinkhorns. The fungus is an organism that is eukaryotic and it possesses cell walls with chitin and they grow by extending filamentous cells called hyphae or by budding. They obtain nutrients by releasing digestive enzymes into the environment to break down organic molecules which are then absorbed. So thus they play an important role in decomposition. They have no chlorophyll. Fungi play a vital role in the environment and they are economically important as a food source. Cells of fungi are quite similar to those of plants, protists and animals. Since they lack chlorophyll, it separates them from plants. Since they do not ingest their food, they are not animals. Most fungi reproduce through the generation of spores. Fungal spores are non-motile and this further separates fungi from protists, which also can produce spores, but if they do, they are often motile. Together with bacteria, fungi are the major decomposers of organic materials in the soil. They degrade complex organic matter into simple organic and inorganic compounds and help recycle carbon, nitrogen, phosphorus and other elements for reuse by other organisms. As molds grow by digesting the organic material, they gradually destroy whatever they grow on. It also causes many plant and human diseases. Let's see in detail about yeast, which belongs to the fungus kingdom. They are eukaryotic single-celled microorganisms and having a size range of 3 to 4 micrometer in diameter. They are spherical to ovoid, lemon-shaped, pear-shaped, cylindrical or triangular. This is the image of a typical yeast cell. It consists of a cell wall, plasma membrane, periplasmum, nucleus, peroxisome, vacuole, mitochondrion, Golgi, bodies and cytosol. Let's see the function of each one. The first one is peroxisome. It breaks down very long chain fatty acids through oxidation. Cytosol. It is the liquid phase of the cytoplasm in an intact cell. This excludes any part of the cytoplasm that is contained within the organelles. It constitutes most of the intracellular fluid. Vacuole. It's an extremely complex organelle. It is involved in a wide variety of functions such as to carry out degradatory processes and is also acting as a primary storage site for certain small molecules and have biosynthetic precursors such as basic amino acids and polyphosphate, and it also maintains intracellular pH. Yeast reproduction can be broadly classified into asexual and sexual, and the asexual reproduction is through budding and binary fission, and sexual reproduction is through the formation of an appropriate spore structure. In contrast to mammalian cells, peculiarities of yeast cells are that they are surrounded by a rigid cell wall and develop birth scars during cell division. The vacuole corresponds to lysosomes in higher cells. Birth scars are specialized ring-shaped convex protrusions at the cell surface which remain on the mother cells after cell division and birth of daughter cells. The concave indentations remaining on the surface of the daughter cells after budding are called birth scars. Most yeast reproduce asexually by budding. In budding, a small bud cell forms on the cell, which gradually enlarge and separate from the mother cells. In binary fission, there is splitting in two that is occurring in the fission yeast. In this image, you can see the development of new buds and the chain of buds that is formed gradually. In budding, the parent cell forms a protuberance on its outer surface, and as the bud elongates, the parent cell's nucleus divides and one nucleus migrates into the bud. The cell wall material is then laid down between the bud and the parent cell, and the bud eventually breaks away. The yeast reproduce rapidly, that is, one yeast cell can in time produce up to 24 daughter cells by budding. An example of the budding yeast is Saccharomyces, and it divides unevenly. You can see that the bud appears and the DNA will double. Later the nucleus will replicate and the nucleus will move into the bud, and the nucleus separate and the cells divides, forming scar at division site. And you can see that after the division, you can see the bud scar here. Some yeast produce buds that fail to detach themselves, and these buds form a short chain of cells called a pseudohypha. Candida albicans attach to human epithelial cells as a yeast, but usually require the pseudohypha to invade into the deeper tissues of the human. The budding only takes place in favorable conditions and when the yeast cells are grown in sugar solution. And it is a form of asexual reproduction in which a new organism develops from an outgrowth or bud due to cell division at one particular site. And the next one is binary fission, that is similar to budding. During this, the parent cell elongates, grows to certain size, its nucleus divides, and two equal daughter cells are produced. Only a few yeast species reproduce by this fission, and an example is the Saccharomyces, and it divides evenly to produce two new cells. Next is the sexual reproduction in yeast. An ascus is a sexual spore-bearing cell produced in ascomycete fungi. Asca usually contain 8 ascospores produced by meiosis, followed in most species by a mitotic cell division. Now let's see the physiological characteristics of yeast. It grows best with a plentiful moisture. Many yeast grow in the presence of greater concentration of solutes such as sugar or salt. It requires less moisture than most bacteria. Most yeast requires more moisture than molds. On the basis of water activity, there are two types of yeast. The ordinary yeast that do not grow in high concentration of solutes, and it can't grow in low water activity food materials. The supporting water activity ranges between 0.88 to 0.94. And the other one is the osmophilic yeast, and it grows in high concentration of solutes. It grows in low water activity food materials and may grow slowly in media with an water activity as low as 0.6 to 0.65 in syrups. It has an optimum temperature of 25 to 30 degrees Celsius and pH range of 4 to 4.5, and it lacks chlorophyll and need carbon source like sugar, and needs source of nitrogen like ammonia, nitrate, urea, protein, peptide, amino acid. These grow best under aerobic conditions, but some are anaerobic. Let's see a few of the cultural characteristics when the yeast are grown. The colonies are usually moist, slimy, dry, or wrinkled. And the colony color can be whitish, cream colored, or pink. Then there are oxidative yeast and fermentative yeast. The oxidative yeast oxidize organic acids and alcohol. They grow at the surface as pellicle or scum in the broth. And in the fermentative yeast, it will ferment glucose to ethanol and carbon dioxide, and the growth is throughout the liquid medium in the case of fermentative yeast. Yeast are facultative anaerobes which allow them to grow in a variety of environments. When oxygen is available, they carry out aerobic respiration, and when oxygen is not available, they ferment carbohydrates to produce ethanol and carbon dioxide. Yeast can use oxygen or an organic compound as a final electron acceptor, and this is valuable attribute because it allows these fungi to survive in various environments. If given access to oxygen, yeast perform aerobic respiration to metabolize carbohydrate to carbon dioxide and water. If they are denied of oxygen, they ferment carbohydrates and produce ethanol and carbon dioxide. And this fermentation is used in the brewing, winemaking, and baking industries. Saccharomyces species produce ethanol in brewed beverages and carbon dioxide for leavening bread dough. Yeasts are chemoorganotrophs, as they use organic compounds as a source of energy and do not require sunlight to grow. Carbon is obtained mostly from hexose sugars such as glucose and fructose, or from disaccharides such as sucrose and maltose. They can metabolize pentose sugars such as ribose, xylose, and can metabolize alcohols and organic acids. Yeast can be grown in the laboratory on solid growth media or in liquid broths. They grow especially well in substances containing sugar. Increase in the number of yeast cells on a solid medium produce a colony similar to a bacterial colony. The common media that is used for the cultivation of yeast include potato dextrose agar or potato dextrose broth, Wallerstein Laboratories nutrient agar, yeast peptone dextrose agar. Let's see the genus Saccharomyces. There are two categories, the top yeast and the bottom yeast. The top yeast are very active fermenters and they grow rapidly at 20 degrees Celsius. The clumping of the cells and the rapid evolution of carbon dioxide sweep the cells to the surface and hence the term top yeast. The bottom yeast do not clump and grow more slowly. They are the best fermenters at lower temperature, that is 10 to 15 degrees Celsius. The absence of clumping and the slower growth and evolution of carbon dioxide permit the yeast to settle to the bottom and hence the term bottom yeast. Saccharomyces cerevisiae variety ellipsoides is a high alcohol yielding variety and is used to produce industrial alcohol, wines, distilled liquors. And Saccharomyces euarum is a bottom yeast and it is used in making beer. And Saccharomyces fragilis and Saccharomyces lactis has the ability to ferment lactose, so they are important in milk or milk products. Then there is Saccharomyces roxy and melis, which are osmophilic that can survive under high sugar concentrations. Then there is genus Zygosaccharomyces, which is osmophilic that can survive under high concentration of sugar. So they are involved in the spoilage of honey, syrups, molasses, soy sauce, wines. Then there is the genus Pichia. These are oval to cylindrical yeast and form pseudomycelia. Here the ascospores are round or hat-shaped and there are 1 to 4 spores per ascus. A pellicle is formed on liquid and the species Membrana faciens grow a pellicle on beers or wines. The next genus is the Devariomyces. These are oval or round yeast and form pellicles on meat brines, and the ascospores have a warty surface, and the species Clerkery grows on cheese and sausage. There are also false yeast that lack sexual mode of reproduction, and that includes genus Torulopsis, Rhodotorula, Candida, Brettanomyces, Clercara, Trichosporum. Torulopsis shapes round to oval and it is a fermentative yeast. It's having multilateral budding and it causes trouble in breweries And Turulopsis spherica ferments lactose and it may spoil milk products. Other species of Turulopsis can spoil sweetened condensed milk, acid juice concentrates, acid foods. Candida. Its asexual reproduction is by budding or chlamydospores and it spoils foods that is high in acid and salt, fruit juices, pickle, and also Candida utilis is grown for food and feed. Candida krusei has been grown with dairy starter cultures for maintaining the lactobacillus activity and increasing the longevity of the lactic acid bacteria. Candida lipolytica can spoil butter and oleomargarine. Rhodotorula cause discoloration on foods like red and pink areas in sauerkraut, yellow, and produce colored spots on meats. Now let's see the property of mold. It belongs to the fungus family and they are multicellular filamentous fungi. They are microscopic and plant-like organisms, and their growth can be easily recognized by its cottony appearance. Molds are very common organisms that grow on food materials to spoil the foods and can be easily identified. They have fuzzy or cottony appearance and their vegetative body or thallus lack true root stems or leaves. Hyphae is a long branching filamentous structure in fungi. There are two forms of hyphae. The septate hypha, this is having a compartment-like appearance, and the coinocytic hypha. Mycelium is a vegetative part of the fungus consisting of a mass of branching thread-like hyphae. Hyphae of most molds contain cross walls called septa, which divide them into distinct uninucleate cells-like units. And hyphae of few classes of fungi contain no septa and appear as long continuous cells with many nuclei. Cytoplasm passes through and among cells of the hypha uninterrupted by cross walls, and they are termed as coinocytic hyphae. Hyphae grow by elongating at the tips. Each part of hyphae is capable of growth, and when a fragment breaks off, it can elongate to form a new hyphae. In the laboratory, fungi are usually grown from fragments obtained from a fungal thallus, or the vegetative body. Portion of a hyphae that obtains nutrients is called the vegetative hyphae. Portion concerned with reproduction is the reproductive or aerial hyphae, so named because it projects above the surface of the medium on which the fungus is growing. Aerial hyphae often bear reproductive spores. When environmental conditions are suitable, the hyphae grow to form a filamentous tangled mass called a mycelium, which is visible to the unaided eye. Molds release countless tiny microscopic cells called spores, which spread easily through the air and form new colonies where they find the right conditions. For molds to grow and reproduce, they need only a food source. It can be any organic material such as leaves, wood, paper, or dirt, and moisture. Development of fungi cultures usually begin with a spore. In the presence of moisture, the spore swells with water, much like a germinating plant seed. Then the spore wall expands through a preformed weak spore to create a thin balloon-like protuberance. This first extension of growth is called a hypha, resembling long, worm-like structures. And with continued growth, the hyphae will branch and grow into a visible colony called a mycelium. Almost all the fungi are chemoheterotrophs requiring organic compounds for energy and carbon. They are mostly saprophytes, that is, they obtain their nutrients from dead organic matter. The fungi play an important role in the environment by decomposing and recycling organic matter. And few of the fungi are parasiting on plants, causing major losses of crops. And others also infect humans or animals and cause serious infections. Molds may grow as loose and fluffy, compact, velvety on the upper surface, or sometimes dry and powdery, or wet or gelatinous. Also, the mycelium may grow colored, like for example, Penicillium notatum it's yellow, Aspergillus flavus it's brown, for Mucor and Botrytis cinerea it's grey, for Cladosporium it's black, and for Penicillium chrysogenum it's blue-green. The most molds grow well at ordinary temperature and their optimum temperature is 25 to 30 degrees Celsius. But few grow well at 35 to 37 degrees Celsius or above, just like the Aspergillus. And a number of molds grow well at refrigeration and freezing temperature, and it's around -5 to -10 degrees Celsius, and they can be termed as psychotropic. And few are thermophilic, that means it can grow at a high temperature. The moisture requirement is 0.6 to 0.65, and it molds require less moisture to grow than yeast and bacteria. If dried food has a moisture content below 14 to 15%, it will prevent or delay mold growth. Molds are aerobic and require oxygen for their growth, but in fungi in general are aerobic or facultatively anaerobic, or it can be anaerobic alone. The pH range of mold is between 2 to 8.5, and the majority are favored by an acidic pH. It can utilize many foods ranging from simple to complex. Most molds possess a variety of hydrolytic enzymes, and some are grown for their amylases, pectinases, lipases, and proteinases. That is, they have been industrially exploited for the production of these enzymes. Compounds inhibitory to other organisms are produced by some molds. For example, the antibiotic penicillin produced by Penicillium chrysogenum and clavacin produced by Aspergillus clavatus. So where are these molds found? They are found in every environment and can be detected both indoors and outdoors year-round. Mold growth is encouraged by warm and humid conditions. In outdoors, they can be found in shady, damp areas or places where leaves or other vegetation is decomposing. In indoors, they can be found where humidity levels are high, such as basements or in showers. Reproduction mold can be both asexual and sexual. Asexual reproduction is the most common mode. The spores are produced in large numbers, they are small and light, they are resistant to drying, and they are readily spread through the air. They grow as new mold thallus under favorable conditions. The first one is the conidia. Conidia are cut off or bud from special fertile hyphae called conidiophores. They are not enclosed in any container, and examples of these conidia found are in Penicillium and Aspergillus. The next type is the arthrospores or oidea. In this, the hyphae breaks up into small pieces and develop into spores. It cannot survive in unfavorable conditions. Next one is the sporangiospores. The spores that are produced in a sporangium, and the sporangium is termed as an enclosure in which the spores are formed. And examples of these type are the rhizopus and the mucor. Next, let's see the sexual spores. Sexual spores result from the fusion of nuclei from two opposite mating strains of the same species of fungus. Fungi produce sexual spores less frequently than asexual spores. Organisms that grow from sexual spores will have genetic characteristics of both parental strains. Sexual reproduction consists of three phases. First one is the plasmogamy or the cell fusion. Here, a haploid nucleus of a donor cell penetrates the cytoplasm of the recipient cell. In karyogamy or nuclear fusion, the positive and negative nuclei fuse to form a diploid zygote nucleus. And the last stage is a meiosis. The diploid nucleus gives rise to haploid nuclei, some of which may be genetic recombinants. The first one is the oospores. They are termed oomycetes. These molds are mostly aquatic and important plant pathogens. The oospores are formed by the union of a small male gamete and a large female gamete. Next one is the zygospores. Zygomycetes form zygospores by the union of the tips of two hyphae. Hyphae may come from the same mycelium or from different mycelia. Zygospores are covered by a tough wall and can survive drying for long periods. Here you can see the formation of zygospores. The next sexual spores is the ascospores. The ascomycetes form sexual spores known as ascospores. It is formed from the union of two cells from the same mycelium or from two separate mycelia. The ascospores are in an ascus or sac with usual 8 spores per ascus. In this you can see the ascae each with 8 ascospores. Common molds include Absiria, Fusarium, Mucor, Penicillium, Aspergillus, Basidiomycetes, Clarosporium, Trichoderma, Ulloclarium, etc. Let's see the industrial importance of Penicillium. Penicillium camemberti consists of a grayish conidia and it's useful in the ripening of camembert cheese. Penicillium roqueforti consists of a bluish green conidia and it aids in the ripening of blue cheese. And an example of the blue cheese is the Roquefort cheese. And from Penicillium chrysogenum we can derive the antibiotic penicillin. Let's see some detrimental effects of the penicillium. Penicillium expansum as a blue-green spored mold and it causes soft rods of fruits. Penicillium digitatum causes soft rods of citrus fruits and it is having a yellowish green conidia. And Penicillium italicum also causes rods in citrus fruit and it consists of a blue-green conidia. The next one is the geotrichum. The species may be white, yellowish, orange or red, with the growth appearing first as a firm fat-like mass that later becomes soft and creamy. Geotrichum candidum is also called as dairy mold and it gives rise to white to cream colored growth. And the next one is the Rhizopus. It is commonly known as the bread mold. It consists of a rhizoid, a root-like part of the hyphae that anchors the funcus, and also consists of a stolon. It's a horizontal hyphae that connects the group of hyphae to each other.